Hello, this is Feiler, and I'm CTO at Nanologica, and today I'm going to speak about NLAB Spiro, the next generation drug carrier for inhalation. Nanologica is a biotech company based in Sweden manufacturing porous silica particles with ton-scale capabilities under GMP. The NLAB Spiro platform is low-density, porous, amorphous silica particles designed for dry powder pulmonary delivery. The particles are manufactured with very tight particle sizes, typically 1 to 5 micrometers, ideally suited for direct lung delivery. The particles are characterized by nanometer-sized pores, typically in the size 10 to 20 nanometers, which results in particles with very high surface areas, typically 300 to 500 square meters per gram. We can encapsulate a range of APIs in the particles trivial loading procedure, resulting in dry powder formulations of difficult to handle APIs. And this gives us a true platform technology, providing a range of APIs for direct lung delivery. The silica particles are amorphous, and this shouldn't be confused with quartz, and amorphous silica is soluble, and in our case, the particles are soluble within 24 hours in lung fluid. Here is an example where we have taken a hard-to-handle API and we formulated it into NLAB Spiro particles, producing a free-flowing powder. In this case, this is a very poorly soluble API, giving crystalline and also sticky material. And when we load it into the NLAB Spiro particles, we have a free-flowing powder. One of the key features of NLAB Spiro technology is the ability to encapsulate drugs in the amorphous form within these nanopores. When crystalline drug is encapsulated in nano-sized pores, it is prevented from recrystallizing, and this results in stabilizing the amorphous form. In addition, the very large surface areas within the particles means that when the particles are exposed to lung fluid, this very large surface area results in rapid dissolution kinetics of the API. The increased dissolution kinetics then leads to an enhanced permeation and enhanced bioavailability of the API. NLAB Spiro particles, as I mentioned, are tailor-made for lung delivery, and we can tune the particles to reach the desired position in the lung. In this figure, in the top, we show where we've taken a commercial API that is very poorly soluble and hard to handle, and we have encapsulated it in NLAB Spiro, and we have shown a dramatic enhancement in the dissolution kinetics. And in the lower graph, we show that the particle size measured by impactor measurements is ideally suited for direct lung delivery, and importantly, the powder flow properties doesn't change when we encapsulate the API. 99% of the surface area is within the pores, so only 1% is on the outside. And this means that when we load the APIs into our particles, all of the API is encapsulated from the inside. And the resulting dry powder is what we then deliver to the lung. Another key feature of the technology is that porous silica particles are mucoadhesive. In this image, we see SEM micrographs of the silica particles slowly becoming embedded in the mucin layer. So we can imagine the application of these finely dispersed dry particles attached to the lung surface and slowly becoming embedded in the mucin layer. The drug dissolution kinetics, of course, is governed by the properties of the drug, and the silica particle is slowly dissolved within 24 hours. And there is no residual particles, and there is no geno or mutotoxicity known to be associated with amorphous silica particles. We have formulated a range of different APIs in NLAB Spiro technology, and in this case I give 3 examples of 3 recent formulations where we've taken existing molecules and formulated them into dry powder formulations. On the left we have an asthma medicine that is suffering poor solubility problems. We have an antibiotic drug that also is very poorly soluble, and we have formulated this into a formulation with dramatically enhanced permeation. And on the right, we have an antiviral drug that we formulated this summer in response to COVID-19. The images show impactor measurements, which is a measure of the particle size distribution, and the particles all fall within the desired particle size range, giving a dry powder formulation with excellent powder properties. Some common problems with dry powder formulations are poor patient-to-patient variability, particularly in diseased lung with impaired lung function, or poor delivered doses due to either losses in the capsule or the device, or getting stuck in the throat and not actually reaching the target site. In our case, we have shown that we have formulated a corticosteroid, and we have got 70% of the delivered dose in the fine particle fraction available for lung delivery. This, we believe, is a real improvement on manufacturing capabilities, giving higher doses and potentially improving patient compliance due to better outcomes. Another challenge with development of drugs for dry powder formulation is the lack of good in vitro models to translate dissolution and permeation into clinically relevant formulations. A commonly used technique is isolated perfused rat lung, and in this case we show example where our formulation with NLAB Spiro has been compared in a back-to-back study against a nebulized formulation of the drug. And in this case we show a dramatic enhancement of the fraction of absorbed drug through the lung. In the nebulized formulation, 20% of the drug is absorbed within 1 hour, whereas in our case, 80% of the drug is absorbed within 1 hour. As an additional benefit to NLAB Spiro technology, we see great potential to offer combination products. In this image, we show a schematic of several APIs loaded into the particles, and the particles can then be combined to a single dosage and then delivered as a combination product. In this way, we can tune the particle concentration to give the desired dose, leading to tailored and personalized formulations. So in summary, I have presented a new pulmonary delivery technology based on porous silica. Nanologica manufactures our porous silica particles at large volumes under GMP. The particles have very narrow particle size distribution, ideally tailored for direct lung delivery. The particles carry a high drug load, typically 30 to 50 weight%, and the drug is encapsulated in the particles, which have very good aerodynamic properties, and the particle flow properties are independent of the loaded API. The silica is amorphous silica, and it is biosoluble, dissolving over a time frame of 24 hours. And similar silica has been studied in animal studies showing no acute toxicity and no long-term toxicity effects. We are currently at preclinical phase, and we have projects with our partners moving into clinical trials. And Nanologica is also developing internal assets, which is moving into clinic. So we invite you to study the technology and contact us with your questions and needs of formulation, and we'll be extremely happy to discuss formulation project. Typically, we begin with a feasibility study, and then we can provide material for testing and evaluation. So please do feel free to contact Nanologica with any questions you have on Spyro technology. If you contact Kia Bengtsson, Head of Drug Delivery at Nanologica, she can answer commercial questions and set up a chance for a feasibility study. And please direct any other questions to spyro@nanologica.com, and we can answer any further inquiries regarding this platform technology. Thank you.